Welcome to Evolutionary Biology. Today, we are going to explore a fascinating phenomenon known as the founder's effect. Have you ever wondered why certain rare genetic traits suddenly become incredibly common in isolated populations? It is not always about survival of the fittest. Sometimes it is simply about who happened to be in the right place at the right time. Let us dive in and discover how random chance can completely reshape the genetic destiny of a brand new colony. To truly understand what the founder's effect is, let us start with a simple thought experiment. Imagine you have a massive glass jar filled with 10,000 jelly beans of 20 different colors. This represents a beautifully diverse original population. Now, suppose you blindly reach in, grab exactly 5 jelly beans, and put them into a brand new empty jar. What are the chances that your new jar has all 20 colors? It is absolutely impossible. Because you only took 5 beans, your new collection will look completely different and be much less diverse than the original one. In evolutionary biology, when a tiny group wanders off to start a new population, they act just like those 5 jelly beans. Now that we have a mental picture from our jelly bean analogy, let us look at the formal biological definition. The founder's effect is a specific type of genetic drift. It occurs when a very small number of individuals break away from a much larger parent population to establish a new colony. Because this founding group is so small, they do not carry all the genetic variation of their ancestors. Instead, their genetic makeup is essentially a random, limited sample of the original population's genes. This random sampling is the core engine driving the evolutionary changes we see in these new isolated colonies. Building on that definition, what exactly happens to the genetics of this new colony? The most immediate impact is a significant reduction in overall genetic variation compared to the parent group. But something even more interesting happens. Certain traits or alleles that were extremely rare in the original population might suddenly become very common in the new one. Why? Simply because one of the few founders happened to carry that rare gene. Over time, as this small group reproduces and grows, the entire new population will reflect the specific genetic quirks of those few original founders, rather than the diverse parent population. So, does this actually happen in real life? Absolutely. Understanding the founder's effect is crucial because it explains why certain isolated human populations have unusually high rates of specific genetic conditions. For example, if we look at the Amish communities in Pennsylvania, or the inhabitants of the incredibly remote island of Tristan da Cunha, we find that certain rare genetic disorders are surprisingly common. These are textbook examples of the founder's effect in action. A small number of original settlers founded these communities, and they happened to carry specific rare genes that were then passed down to thousands of descendants. Looking at these real-world examples brings us to a profound realization about evolution. If we did not understand the founder's effect, we might incorrectly assume that these common traits in isolated populations were somehow beneficial and favored by natural selection. Instead, we know it is simply a matter of chance. One of the original founders happened to carry the gene, and because the population grew from such a small starting point, the gene became widespread. It beautifully illustrates that evolution and genetic change can happen purely by random chance, completely independent of any survival advantages. Let us solidify this concept by looking at a visual representation. On the left side of this diagram, you see the original population, represented by a large circle filled with dots of many different colors. This shows high genetic diversity. Now, follow the arrow showing the migration of a small group. This tiny founding population only contains red and blue dots. They have left all the green, yellow, and purple genetic variations behind. Finally, look at the new population on the far right after many generations. It has grown in size, but it only contains the red and blue traits of its founders. The diagram perfectly captures how a bottleneck event permanently strips away genetic variation. Now that we understand the concept visually, let us look at the mathematics behind losing genetic diversity. When a new population is founded, we often want to know the probability that a specific genetic variant, or allele, is completely left behind. This is given by the formula P loss equals 1 minus P, all raised to the power of 2NF. Let us break down these symbols. P loss is the probability that the allele is completely lost. The variable P represents the frequency, or percentage, of that specific allele in the original large population. NF stands for the number of founding individuals. We multiply NF by 2 in the exponent because we are assuming these are diploid organisms, meaning each individual carries 2 copies of every gene. If the number of founders increases, the probability of losing the allele drops dramatically. Conversely, if the allele frequency P is very small, it is highly likely the rare allele will be left behind. Let us apply this formula to a concrete example. Imagine a mainland bird population where a gene for blue feathers is rare, with a frequency of just 5%. So P equals 0.05. A storm blows exactly 4 birds to a remote island, so our number of founders, NF, is 4. We want to find the probability that the blue feather gene is completely lost. Because the birds are diploid, 4 founders carry a total of 8 alleles. We substitute P equals 0.05 and NF equals 4 into our equation. The term inside the parentheses becomes 0.95, representing the 95% chance that any single randomly chosen allele is not the blue feather allele. Raising 0.95 to the power of 8 gives us approximately 0.663. This physically means there is a 66.3% chance that the new colony will never have blue-feathered birds, demonstrating how easily rare traits are lost. We just calculated the probability of losing a single specific trait. But another way scientists measure the founder's effect is by looking at the overall genetic diversity of the entire population. In evolutionary biology, this overall genetic diversity is known as heterozygosity. By comparing the heterozygosity index of the newly founded population to the original parent population, we can quantify exactly how much genetic variation was lost across the board, simply due to the small sample size of the founders. This gives us a broader picture of the genetic health and diversity of the new colony. To find this new overall genetic diversity, we use the heterozygosity formula. The formula states that H1 equals H0 multiplied by the quantity 1 minus the fraction 1 over 2NF. Let us define these terms. H1 is the new genetic diversity of the founder population in its very first generation. H0 is the original genetic diversity index of the large parent population. And NF, just like before, is the number of founding individuals. The fraction 1 divided by 2NF is particularly important. It represents the exact proportion of genetic diversity that is expected to be lost purely due to the small sample size of the founders. As the number of founders, NF, increases, this fraction becomes smaller, meaning less diversity is lost. Let us see this heterozygosity formula in action. Imagine a population of mainland lizards with an initial genetic diversity index, H0, of 0.80. A small group of just 10 lizards floats on a log to a new island, so NF equals 10. We want to calculate the expected genetic diversity, H1, of this new population. We substitute H0 equals 0.80 and NF equals 10 into our equation. The fraction becomes 1 divided by 20, which is 0.05. This means 5% of the genetic diversity is lost immediately. Subtracting this from 1 leaves 0.95, meaning the new population retains 95% of the original diversity. Multiplying 0.80 by 0.95 gives a final expected genetic diversity of 0.76. Physically, this shows how a bottleneck event permanently strips away genetic variation in a single step. We have covered a lot of ground today, so let us summarize the core takeaways. The founder's effect is a powerful evolutionary force that demonstrates how random chance can shape the genetic destiny of a population. Whenever a small group breaks away to start a new colony, they take only a tiny, random fraction of the original genetic diversity with them. This inevitably leads to a new population that is genetically distinct, less diverse, and often prone to rare genetic traits becoming surprisingly common. Ultimately, this phenomenon reminds us that survival of the fittest is not the only way populations evolve. Sometimes, evolution is simply about who happened to be on the boat.